Welcome back, everyone, for our final sessions of what has really been an inspiring meeting. In this next session, we'll have two final plenary talks describing forward-looking collaborative international efforts that are already bringing together the community to make discoveries. We maybe somewhat audaciously titled this session, The Future of Genetic Maps. It'll be moderated by Mark Daly, who is the director of the Institute for Molecular Medicine in Finland, as well as an institute member and co-director of the Medical and Population Genetics Program at the Broad Institute, and the founding chief of the Analytic and Translational Genetics Unit at Mass General Hospital. Mark, take it away.
Thanks, Rachel. Hi, everyone. Really fired up to introduce the final plenary session of the meeting. It's been a fantastic meeting, and glad everyone has stuck around to what will be exciting rather than bitter end of the meeting. The MAPS aspect of the Mechanisms to Medicine, has been focused on the identification of where resources are needed that would promote research broadly across all common diseases, all rare diseases, but broadly even beyond the boundaries of any ICDA that we define. And of course, those resources include the construction and ability to make powerful and available global variation patterns and in a way that's as comprehensive and as useful as possible. And also resources such as the GWAS catalog to make the results of GWAS and rare variant sequencing studies as broadly available to the entire world as possible. And an effort to, sort of building from what you heard earlier from Andres, Ricardo, and many, many others, the effort to build biobanks around the world is a crucial one for ICDA, but really for, human genetics, biology, and medicine in general. And in order to make the best, of emerging resources and to support those resources in building a greater universal whole, we in the MAPS working group have also been focused on how do we construct the infrastructure and the frameworks into which biobanks around the world can work together in a fair and equitable way to advance our knowledge of disease, common and rare. And so we have a couple of talks that I think exemplify some of the things that we'll be aiming for in the future in ICDA and beyond. And the first talk today comes from Wei Zhou on behalf of the Global Biobank Meta-Analysis Initiative that was launched in 2019. And this will describe collaborative efforts of biobanks around the world to power genetic discovery. Welcome, Wei. Great to have you here.
Thanks, Mark, and the ICDA organizers for the opportunity for me to present our collaborative efforts of biobanks to power genetic discovery across human diseases on behalf of GBMI. So Global Biobank Meta-Analysis Initiative, as Mark mentioned, was started in early 2019 by Mark, Ben, and Kristen Willer, with the goal to bring together large-scale genomic studies to foster collaboration to understand the genetic basis of human diseases. By aligning phenotype definitions and analysis methods, we hope to gain insight into associations shared across different ancestries and environments. After several months discussion, we have finalized our governing principles of collaboration to ensure a collaboration environment with honesty, fairness, and trust, as well as respect. So far, we have 20 biobanks with different origins and ancestries join GBMI, bringing our total number of genotype samples to be larger than 2.3 million. So here in this map, we are listing all the biobanks in GBMI, coloring them by continents. As we can see, we have 2 biobanks in East Asia and 1 biobank in Australia, 8 biobanks in North America, and 9 biobanks in Europe. So although we know biobanks from North America and Europe are predominated by samples with European ancestry. And we do have biobanks contain non-European samples, such as MVP, BioMe, BioView, UCLA, East London Genes and Health, as well as UK Biobank. And we know biobanks usually have different sampling strategies to recruit samples. So here in this map, we're coloring them by different sampling strategies. And we see several biobanks recruit general population. So they're population-based biobanks, and other biobanks are hospital and health center based. And several biobanks use both strategies to recruit samples. Biobank meta-analysis can have many potential benefits. I'm listing several of them here, including the increase in ancestry diversity of genetic studies, and we hope to get better power of GWAS. It also gave us the opportunities to study some less prevalent diseases that have been understudied previously. And we hope to gain more accurate PRS, improved fine mapping, and more interestingly, we want to create a collaborative network that allow subgroup projects among biobanks based on the research interests in common. To demonstrate these benefits and how GBMI works, we're currently working on a flagship project in which biobanks shared some GWAS summary stats for 14 endpoints. So they're listing here ranked by their prevalence from high to low. When we select endpoints in this list, and we select some common diseases that have been well studied by previous GWAS, so we can compare all the results to those GWAS. And for example, asthma is selected here. And we also select some less prevalent diseases, such as abdominal aortic aneurysm, idiopathic pulmonary fibrosis, thyroid cancer, and cardiomyopathy. We select a female-specific phenotype, uterine cancer, and also we include a procedural endpoint, appendectomy, as well as the associated diseases, acute appendicitis. For the flagship project, we follow the unified analysis pipeline. Firstly, we harmonize our phenotype definitions across biobanks. Next, each biobank runs GWAS to generate summary stats by ancestry and by sex, if possible, by phenotyping approach. After QC, we conduct all biobank meta-analysis as well as leave one biobank meta-analysis for each endpoint, which might be very useful for individual biobanks to construct PRS for those endpoints and to conduct other analysis. And following meta-analysis, a variety of downstream analysis are conducting in order to evaluate heterogeneity across biobanks and studies, to dissect the novel association hits we have found, and also to demonstrate benefits from biobanks working together. So this is a huge teamwork, including analysts from individual biobanks, as well as the centralized analysis team. We have generally observed different disease prevalence across biobanks. I'm listing 4 examples here. On the left, we're showing prevalence. On the right, we're showing the number of cases. The biobanks are colored by their sample populations. So we do see GBMI have lots of non-European samples in the study. While following the harmonized phenotype definitions, biobanks are encouraged to construct phenotypes by making the best use of the available health data. Thanks to the Pan-UK Biobank team, which has run GWAS on thousands of phenotypes on the entire UK Biobank, including non-European samples that are led by Conrad and Alicia. So we're using asthma in the UK Biobank as an example. Here we're plotting number of cases for the 6 different ancestries in UK Biobank. and based on different phenotype definition, including ICD codes mapped to phe code, self-reported, either or both. So as expected, we see different number of cases according to different definitions, and we generally see quite high genetic correlation using those different definitions. But this need to be evaluated more for more phenotypes and more biobanks. Another interesting experiment we want to try is to evaluate genetic ancestry of participants in different biobanks in the same PC plots via PC loading sharing. So what we're doing is Alicia has conducted PCA in the reference samples, including 1000G and HGDP, using 190,000 LD prone genetic markers that are observed in all GBMI biobanks. So once we generated the PC loadings, we share those loadings for each marker back to individual biobanks in GBMI. And so individual biobank analyst who has access to the raw genotypes of their participants can then generate PC scores for their participants and share back for those PC scores. So then we can try to plot the individual biobanks participants based on their PC scores in the same PC plot. Here we're using 3 biobanks in GBMI as an example, and Masaknai has constructed PC scores for Biobank Japan's participant, which are represented by the brown dots. As we can see, they are perfectly located in the East Asian reference samples. And next is the Hunt study from Norway, and the samples are located in the with the European reference samples. And as you can see, UK Biobank has samples with multiple ancestries, so the blue dots spread everywhere. And we can then take a look at the GBMI samples only. Although we only include 3 example biobanks here, we do see GBMI incorporate samples with diverse ancestries in these studies. So next is based on 16 biobanks GWAS results, and we can conduct meta-analysis for asthma and containing 145,000 cases and over 1.5 million controls. So here's a Manhattan plot for meta-analysis results. We have identified 139 loci that are past the genome-wide significance threshold, in which 44 are potentially novel. And the pie chart shows us the proportion of the population of the samples included in the meta-analysis. As you can see, over 20% of our samples are non-European samples. So this allows us to use the genetic analysis results to validate the integration of association results across biobanks. So what we have done is we extract 27 asthma index variants from multiple previous GWAS for asthma. And we use 19 out of them, which are biallelic and have effective sample size over 10,000. And on the left, we plot the effect sizes for those index variants from all biobank meta-analysis, as we see the y-axis represents, and compare them to the previous GWAS effect sizes as the x-axis represent. So each dot here is an asthma index variant, and we generally see consistent effect directions, but attenuated magnitude of those effects in all biobank meta-analysis. And on the right, we're plotting the p-values for the same index variants in meta-analysis and GWAS and previous GWAS. So we do see most of those index variants have more significant p-values in all biobank meta-analysis compared to previous GWAS, indicating larger sample size leading to greater power to detect the association. However, even though we have much larger sample sizes in biobank meta-analysis, and we do see some of those index variants have comparable p-values in the two sets of data sets. And it might be due to several different reasons. One is the potential winner's curse in previous GWAS, or we're using mismapped original index variant reported by previous GWAS. And it is also possible that it's because of the phenotypic variability of asthma across biobanks that may attenuate our association signal in the all biobank meta-analysis. So we then move on with all biobank meta-analysis for all 14 phenotypes. So as we can see here, we have successfully replicated previous reported loci and identified potentially novel loci, as the red bar showed us. And we sharing of the sex-specific GWAS results from biobanks allows us to detect loci with different effects in males and females. So we're showing them, highlight them in red in this plot. Also, we want to account for heterogeneity of genetic variants effects in different ancestries as we incorporate multiple ancestry samples in our meta-analysis. So by accounting for the heterogeneity, and we do identify additional loci on top of the loci that have been identified in the fixed effect meta-analysis. And then we want to plot the most significant variant in each locus in our all biobank meta-analysis. So in this plot, we're plotting the effect sizes versus the risk allele frequency of those top hit. They're colored by phenotype, and the different shape indicator indicates whether the locus is potentially novel for the phenotype or previously reported. So as we can see, the all biobank meta-analysis loci spread over the entire allele frequency spectrum. And we also see some top variants have relatively low frequencies and large effect sizes. And the very top one is HDAC9 for stroke, which is a well-known locus for stroke. Next one, TACC2. This gene has been previously shown to be associated with lung cancer. So we identify this locus for IPF, idiopathic pulmonary fibrosis. And this gene has also been recently shown to be differentially expressed in IPF patients and controls. The next interesting example is the TTC28. We identify this locus for uterine cancer, and this gene has been previously shown to be associated with the ovarian cancer through a whole exome sequencing study. And next, we Huang Wu have run the DPICT to try to run the enrichment test to detect functional tissues and cell types that may be implicated by the associated loci we have identified from the all biobank meta-analysis based on gene expression data. So in this table, we're listing top enrichment tests for the phenotypes that have the signals from the enrichment test with FDR less than 0.2. So we do see some familiar and expected results such as T lymphocytes for asthma and the aortic valve for POAG. But at the same time, we do see some unfamiliar results, and we will need more further investigations on these preliminary results. And lastly, this is a table that we're listing genes that located at a potentially novel loci from our meta-analysis that have one or more coding variants reaching genome-wide significance threshold. So in the interest of time, I'm not going to go through each one of them. And if you spot any your famous gene and have potential hypothesis for the phenotype, please contact us, and we're happy to take any input. So in summary, and biobanks working together can have many benefits, including increase in ancestry diversity of genetic studies. And we hope we use the flagship project to show this, as well as we can get better GWAS power, and we can demonstrate the opportunity to study some less prevalent diseases that haven't been studied by previous research. And also ongoing work includes that we want to show this kind of efforts can help improve PRS prediction and improve fine mapping, as well as create the network to encourage more collaboration among biobanks. We welcome more biobanks to join us, and please visit our website for more details and email us with any question. Huge thanks to all biobanks and biobank participants.
Thanks very much, Wei. That's a fantastic talk and really exciting progress to see. Now we've got some questions that have come in on the Slido, if that's the right way of pronouncing that. First question that came in from an anonymous source is, what sort of reference panel was used to compute or perform the downstream fine mapping activity? Of course, you need an LD reference. And how big do you think the impact of LD differences is on this activity?
Yeah, so Massa Klein is mainly conducting the fine mapping for GBMI, and we basically need to investigate more about the what LD reference panels are going to be used for fine mapping and also for PRS construction, because in our meta-analysis we include all different samples with different ancestries. So that's ongoing work, and we're still investigating.
Next question comes from Ricardo Verdugo. He asked, are the effects from the meta-analysis that you were showing for each specific ancestry, or are they cross-ancestry effects? And he wonders whether the ancestry interactions could explain some of the reduced effect sizes that you saw.
Yeah, that's a great question. So those effects are from the cross-ancestry, and they're from the fixed effect meta-analysis by assuming the effects are the same in all ancestry. So we're going to evaluate the sex-specific effects of those index variants, and also try to expand our analysis to genome-wide through the genetic correlation estimates.
All right, we've got a few more questions that we should try to answer, but I think we need to move on to the next talk in the interest of time, and we'll send some answers to those questions in the via some other meeting. Thank you, Wei, very much. All right, and it's now my great pleasure to introduce Andrea Ganna, who will describe the great activities that have sprung up this year with respect to the COVID-19 host genetics initiative, and sort of unlike the global biobank activity, which many of us have been studying many of these diseases and building biobanks for years, here is a real-time effort to deploy the experience and the vast, comfort and community, good feeling about phenotype sharing for genetic meta-analysis in a real, rapid way to respond to the global pandemic. So welcome, Andrea.
Thank you, Mark, and thank you for inviting me. So I'm speaking on behalf of the COVID Host Genetic Initiative. I'm always very lucky because I'm invited to give this talk, but I'm really speaking on behalf of many scientists that have contributed. I'm going to do a little bit about the science and a little bit about the sociology of putting together such a consortium. So the idea started back in March 2020, as many things today actually start with a tweet. This was the original tweet, and originally we were mostly 3 investigators from Finland, US, and Italy, which thought we would put the data together. And since then we have grown quite a lot, and I will describe it later. But a fundamental aspect was that in the beginning we immediately got support from ICDA, and that was key in order to be able to scale our effort. We got support both in terms of tools, but also administrative and logistic support. So when we set up this initiative, we had to decide what we wanted to do, we focus on creating an environment to sharing resources and to organize analytical activities. This was key. We didn't want just to do a consortium which final aim was a publication, but rather creating an environment that will foster other groups to come together and being able to create projects and create research activities from this platform. Now, we also aiming to create a platform to share the result of such activity. We have been successful in the extent that we were able to get everyone to share summary statistics. We have been unsuccessful in the extent where everyone would share individual level data, which was our original idea. And so it was great to see discussion previously about platform, because there are clearly many challenges in sharing the individual level data at scale. This is shamefully copied from the Global Biobank meta-analysis. Have you seen this slide before? I just want to highlight point number 7, that we should not inhibit any work being done within any individual studies. This was very important for us and a key concept in order to keep the consortium working. I will describe later some of the tension that you have when you have so many studies involved. So as initiative in the beginning, we were not so much focused on doing the GWAS, doing the meta-analysis, but rather to provide the resources. And so in the beginning was clearly key to provide questionnaires so that many studies could initiate their collection. It was very important to provide protocols. And we had done that in different languages, because many of these studies, we didn't actually know how to set up the study in their center, and what was the research question and which kind of legal framework they had to use. We also set up a comprehensive data dictionary with more than 200 variables, and Stefano Ceri at the Polytechnic of Milan has been leading that, which was really key for then some sub-study to be able to come together and collect a uniform set of variables. Now, very important is the communication resources that were fantastically led by Kumar and Brock, not only because it's important to do communication per se, but because communication allowed many people that want to be involved in the consortia, but they do not have data, for example, to find a way to contribute. And so we have seen, for example, that many people have contributed in translating blogs in their own language, and all these people will, be part of the consortia at the same level of other people that have contributed data as well. Because we had this system where study had to register on the website, and I recommend you to go on the website, we had created this catalog of research questions, and you can actually go on the website and contact directly the PI for each study if you're interested about the study per se. Now, most scientists are interested in the meta-analysis result, which are described later, and we have pushed this forward, not just providing the GWAS result, but providing the results from in silico follow-up analysis. We've given the group 48 hours to take the GWAS result and produce in silico follow-up analysis, and many have been able to do that. This is the latest contributor. We have a next data freeze on the 15th of December, so approaching. This is the previous data freeze, but you can appreciate how many of the contributors are from traditional biobank, very similar to the global biobank effort, but then we have group of studies coming from a more clinical environment, and this is very key to contribute, for example, with severe cases. And we have seen direct-to-consumer genetic company, sometimes competing in the customer market, coming together and collaborating and sharing data for this scientific project. We have 36 studies from 16 countries. Now we just clean up the number of registered studies that have confirmed to be willing to contribute. And you see this is 27%, meaning that there's still a lot of potential from other studies and they're still collecting data. The studies are not so well distributed across the globe and reaching US and Europe, but we start to see more contribution coming, for example, from Asia. We have study from the Middle East, from Qatar, for example, and study from Brazil as well. And together in the largest analysis, we have 30,000 COVID positive cases. Now, an important aspect of this initiative is that we wanted to keep open, collaborative, and transparent. The first things we did was to immediately do result sharing, and the result sharing was done and is done, and we have done it now for several releases before publication. This is clearly a fantastic idea and we really support it. Create also some struggle if you think about it, because if we made the result available, then everyone can, for example, publish the result on exactly the GWAS result. So it's a fantastic concept, but clearly we had to think a process in how this result could be used. And so this was a very interesting process, and then there are some guidelines that we have published on our website. The key decision has been voted by the larger community, and this was also important. And then, as you can imagine, in such a large consortia, there is a tension between the larger study that want to go ahead and just use the, let's say, the smaller study as a replication, and the smaller study that clearly want to keep the consortia to advance as for example, for a publication. And so we had to find a way to deal between these two tension, and that was also very interesting, and I think we've been successfully doing that. Our model is that we not inhibit any study, so larger studies can naturally publish their result and use our as replication as much as they share the summary statistics with the consortium. The manuscript writing was also, and is still ongoing, was also done in a collaborative way, so that we accepted community comments to draft an outline, and then we had 6 groups writing each a part of the paper, and then now we are in a phase with just a smaller group putting together all these different parts. Another important aspect is that how do we publish something that changes every 6 months or less? Doesn't make sense to to put out a paper every 3 months if these are disconnected to each other. So we've been discussing with the journal, and the model we've been envisioning, it's actually pretty novel, and assuming that the paper passed the peer review process, then we will be able to repurpose a publication form which was actually originally used to comment or criticize the original paper, by using to just update the paper in every 6 months, for example, so that you can follow the history, you can follow how the result has been updating in a uniform way. And another important aspect is that we're going to do peer review, open review before submitting to the journal, so that this open review will also be submitted with the paper. Now, coming a little bit more to the scientific discoveries. So we focused in the beginning on many different phenotypes. They had to be simple. This was key. We finally converged on these 2 phenotypes as being those that yield the most results. Analysis, what we call analysis B, is hospitalized confirmed cases versus population control, and what we call analysis C is laboratory confirmed cases, self-reported cases, electronic health record confirmed cases versus population controls. Now, using population controls is rather a spurious approach. We know that many of the people in the general population has not been exposed to the virus, so maybe if they were exposed, they will actually end up in the hospital. But we found that this is a more powerful approach, and actually the analysis where the cases have a more appropriate definition of being just asymptomatic, it turns out to have less power and yield less result than doing this population control approach. This is the result of the GWAS. There is a big peak on chromosome 3, which has been previously reported by the hostage team, and I will describe later a little bit more about the following up of this locus. And we have several new loci that I will describe. I just want to highlight how we could really improve the power of this meta-analysis by including the result of the genomic study, which is a fantastic study led in the UK by Kenny Daly, which really focused on the severe cases, so the severe part of the spectrum. And this has yielded quite a lot of results, because really we can see that by focusing on those severe cases, we gain power. Now, in terms of partial susceptibility, you can see that there are signals that are shared. We are on chromosome 3 and on chromosome 9, that's the ABO signal. But there is another signal on chromosome 3, which is specific for susceptibility. Now, the chromosome 3 locus, this is quite fascinating as a quite distinct distribution across the globe. Is not seen in African population, as around 8% frequency in European, and as around 25% frequency in Bangladesh and Pakistani population. Since it's a very recent haplotype, which is obtained through introgression with Neanderthal, has a quite long, there are many SNP in LD, which make quite difficult to do any type of fine mapping. David Van Heel has a very nice study of Bangladesh and Pakistani population in London, and could actually show some of the frequency distribution. So among hospitalized COVID-19 patients, the frequency of this haplotype was 38%. Among those antibody positive but not hospitalized, that was 20%, and among population controls who are not cases is around 27%. You can clearly see the quite a large frequency difference, and you can see how using population control is a little bit more spurious, but it still provide a robust analysis. Now, the first things everyone does when there is a new signal is going and checking if it's associated with something else. And if you take you go to OpenTarget or other FIWAS browser, you will see that this variant is associated with many monocyte measure. However, it's important to think about the co-localization of the signal, as similar to what we do, for example, when we look at EQTL. That applies the same on phenotype. So is actually the peak is the distribution of these associations, similar for phenotype A and phenotype B. And what we actually found that there's actually no co-localization between this signal and these other monocyte GWAS. So this variant associated with the chromosome 3 locus does not end up in the credible set for the GWAS of monocytes, and really make the point that this variant until now is not really associated with anything else than COVID. What we know from the epidemiology of our analysis of the epidemiological part is that these variants is associated with severity, and you can see clearly in data from DECODE where the association is around an odds ratio 1.6 when we compare hospitalized versus population controls, and it dropped to almost 1 when we compare mild asymptomatic versus population control, and an odds ratio of 2, although very large confidence interval, when we look at hospitalized versus mild asymptomatic. Now, let's take now only hospitalized individual. This is a collaboration where we've been able actually to combine individual level data from several studies. When you look only on hospitalized patients, those that carry this haplotype that have around 2-fold higher risk of ending up needing mechanical ventilation. This RESP severe is a mechanical ventilation. This is quite a strong effect. However, we didn't see effect for any other complication beyond just respiratory complications. So there was not a higher risk of any of these other complications among the hospitalized patients. As a consequence that these patients are more severe, they tend also to die earlier. And here you can see that patients carry this haplotype that have around 60% higher risk of dying for COVID-related death. Again, very large confidence interval, but you can appreciate there is a difference in these Kaplan-Meier curves. Now, moving from chromosome 3 to other findings, there is another signal on chromosome 6, which is close to FOXP4. Interesting, this variant is associated with lung cancer in the same direction, so increasing of lung cancer. This mutation is rare in European, but is more common in East Asia and in Hispanic Latino. These really highlight, once again, the importance of including a diverse population. On chromosome 12, we have an association in a locus that contain many of these genes that encode for antiviral restriction enzyme activators. In some single cell analysis, compare COVID severe cases versus non-severe cases. Actually, these genes have the highest differential expression, and this variance is also associated with other phenotypes. Now, this signal is quite interesting, close to TYK2 gene. Why we are thinking this is the missense variant causing the signal is not because we immediately found the missense variants to be the strongest association in the region, but rather because after we impute the variants that were missing, because one of the issues we have with having so many studies contributing is that each of them use a slightly different imputation panel and they use different quality control, and this is create a lack of consistency or heterogeneity in which variants are actually reported. And so we are working to try to reimpute basically this data or this summary statistic to have a better coverage of the region. If you do that for this region, you will see that the strongest signal is for this missense variant, which is protective for these diseases, targeted by drugs. However, in our case, it's increasing the risk, so it goes in opposite direction. Another variant in DPP9 associated with interstitial lung disease, and you can now appreciate two main patterns, a group of variants associated with immunity and another group of variants that seem to be more associated with lung biology or lung function. And finally, a variance in the interferon and the IL-10RB region. Now, ABO stimulates the imagination of many scientists for some reason, and so we have been looking very deep into that. And you can see that we find an association with the hospitalized analysis, so with severity, but with similar effect also with susceptibility. If we look at biobank, which normally enriched for mild cases, with clinical study, which normally enriched for severe cases, and with direct-to-consumer biobank, which normally enriched for self-reported cases. Okay, in conclusion, we work hard to provide a high confidence of association between these loci and COVID severity and partial susceptibility. We focus really on creating robust results that can last the judgment of time. Looking at severe cases versus population control is certainly the most effective design right now. It's pleasant to see that some loci point to clear biology, some lung-related and some other immune-related. But importantly, and maybe one of the most important message from this endeavor is that really by working together as an open community, we can allow for robust real-time replication of findings at an unprecedented pace. So this is something that was achieved in 8 months. And it was inconceivable before the pandemic to put together so many studies at such a fast pace. Concluding by acknowledging many people that too many to put on a slide. You can use this QR code that brings you directly to our website. You has been doing a fantastic job on meta-analyzing all these results together. Okay, thank you.
Thank you, Andrea. That's really fantastic. I mean, it's just been tremendously exciting for me and many of us participating in it to see how quickly everyone in the community has responded and how across biobanks, hospital and critical care studies, and direct consumer companies all have participated enthusiastically in this activity. So we could have some questions now. There's one on the Slido, if I haven't missed more. Now maybe a few actually. So more people were hospitalized and died early in the pandemic than now. And do you think this might have added some noise in the analyses of severity?
Yes, for sure. Speaking, for example, with Italian collaborator, was clear that during the peak of the pandemic, those hospitalized were way more severe than those that are hospitalized now. Some good thing is that we are now collecting information on the period where the sample were collected. And if we will have enough power, we will be able to stratify our analysis later on, and it will be fascinating to see if we see different signal or if the irritability change across, but time will tell.
Any good drug targets coming out of the analysis so far?
I don't know. I don't think so. I mean, the interferon seems promising, but the interferon trial has failed, for my understanding.
Yeah, no, and I think just that's a great question, and really the target of the whole activity, and another great reason why everyone is keen that we make this data immediately available to everyone, because certainly Andrea, myself, and those of us on the genetic side are not necessarily the best people to discern whether something is a good drug target. All right, well, I think we're a little short on time, so thank you very much, Andrea. That was a fantastic talk, and really great to see this work move ahead. And I think it's a great example of what we hope the ICDA can promote and accomplish as a community, and how much more we can accomplish as a global genetics community than individually. So with that, I am happy to turn it back over to Rachel for the final words of the meeting.
Thanks so much, Mark, and to Wei and Andrea for these great presentations. Certainly, I can say it's been very gratifying for ICDA to be involved in the COVID-19 Host Genetics Initiative this past year, and hopefully we'll continue to see more and more results being shared as part of global collaborative efforts like these going forward.